The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, this is episode 80 of the eLife Podcast. I'm Chris Smith from The Naked Scientists. And this month, how COVID antibodies compare between vaccination and infection, baboons show handedness when they communicate, and a new armoured dinosaur. But first, people often say that time rules our lives. But time, it turns out, also rules the pancreas, because the function of the body clock, or circadian rhythm, is tightly linked to the release of the glucose control hormone, insulin. And by tracking the two systems, Joe Bass at Northwestern University has been screening drugs to find molecules that can manipulate insulin levels by tweaking the action of the clock.
A number of years ago, we began to investigate the connection between the body's internal clock and organs in the body that respond to the clock and are important in maintaining energy balance and metabolism. And one of the organs is the pancreas. So a surprise for us was the discovery that the very same mechanism that causes us to wake up each day also induces the pancreas to produce insulin, which controls blood sugar. And so the real purpose of our work has been to try to understand at the most basic level, what are these connectors that links the sleep cycle to the release of insulin?
And I suppose it's not a coincidence then that we know that diabetes or at least an exacerbation of diabetes and sleep deprivation / when people sleep at the wrong times and eat the wrong things, they're all interlinked then.
That's right. We know from epidemiologic studies that there's an association between things like shift work and even exposure to blue light at night, which occurs when you spend a lot of time on computer screens. That kind of light triggers the brain to think that it's daytime even when it's night, and it scrambles the signals internally that normally align our sleep cycle and all of our physiology with the light-dark cycle, with the rising of the sun. And one of the key things that gets scrambled is the ability to metabolize sugar.
So is it that the pancreas knows the time and is doing this itself, or is there literally a connection between what the brain is doing and what the pancreas should be doing?
So the problem, like many things in science, doesn't have a simple answer. Both connections are present. In other words, under normal conditions in evolution, in the context of the wild, so to speak, normally the brain sends signals to the pancreas that entrain the pancreas to function at the right time each day. However, the pancreas has its own internal clock. We believe that one of the things that happens with, say, shift work is that it's the alignment between that brain clock, which responds to the light cycle, and the pancreatic clock that become misaligned. So if we understand how the clock works within the pancreas, we may begin to identify pathways that we could manipulate to realign or fix the broken clock.
And how did you go about doing that?
Well, the first thing is we have to have the tools to measure both the clock and its output. And so we use genetic approaches to insert what's called luciferase, which is an enzyme that's normally made in fireflies. We took a small piece of that gene and stuck it in the middle of insulin, which is the major hormone produced by the pancreas that controls blood sugar. Now, when we do that, that means that every time the pancreatic cells, the beta cells, are stimulated to release insulin, and they are stimulated primarily by glucose, but also by other metabolites and molecules. Every time the cell is stimulated, we have an instrument that can detect the release of the luciferase enzyme. So instead of having to measure insulin, we measure luminescence from this luciferase.
And how do you line that up with what the clock is doing?
So we take another approach using genome editing to go into the genes in the beta cell and twist around the core genes that control the clock, either eliminate or alter the function of the clock mechanism. And with those two instruments, we can now, in a very high throughput way, screen thousands of compounds to ask, do any of the drugs that we have in our libraries influence the release of insulin when the clock is not working?
Did you find any?
We found a number. We screened about 3,000 known drugs. Among these, we identified a handful, and then we used a number of criteria to further select those that we would advance to studying in the animal.
Any that are in common parlance or any we'll be familiar with?
One of the main hits that we had is a somewhat infamous drug at this point. It's called ivermectin. It is an antiparasitic drug, and it acts on a special ion channel in an infectious organism that is unique to that animal, but it turns out that it's in a class of what we call metabotropic receptors, and those receptors turn out to be abundant in the pancreas. And so, for reasons that we don't entirely understand, this molecule is activating a chloride channel that's expressed within the pancreas, and that chloride channel is an important target of the clock. That was a surprise for us to find.
So the chloride channel is affected by the clock, and that in turn affects the secretion of insulin. So therefore you've now got a molecule that can affect the output of insulin from the pancreas when the clock goes wrong. So if you've got people who've got sort of clock-related metabolic syndrome, you could potentially give them this. It might put things right.
What we would do is use this as a so-called scaffolding and to give us molecular insight both into the kind of chemistry that could be used to target the pancreas and also those molecules expressed in the pancreas that are regulators of insulin release. And so that latter idea that this set of receptors are insulinotropic, which means that they induce the release of insulin, is something that we found to be interesting. And there's evidence in the literature for this, and there's also evidence that in obesity, for instance, there may be substances that are produced in fat that interfere with the function of this particular channel. So it's an interesting class of factors that control insulin that we stumbled upon, and they appear to be controlled by the clock system.
Joe Bass. Humans are unique in using speech, and the shapes of our brains reflect the fact that on the left side is a bulge corresponding to the generation and decoding of language. The fact that most people are also right-handed, meaning their left brain hemisphere is dominant, has led researchers to speculate that language goes hand in hand with handedness. And that struck a chord with Adrian Megadichian early on in his career when he was studying our close relatives, the baboons.
We watched the baboons making communicative gesture. And I remember having these baboons come towards me and start to threat me. He was a young baboon and he was slapping his hand towards me and looking in my eyes and repeatedly slap his hand. And I was not moving away, I was just staring at him, and he was keep doing it. And his message was not working, obviously, because I was still around. And he was trying to shake his head, stand up, and all this display was very silent. He was only using his hand, his body posture, to try to make his point. And so the study starts from there. I remember for my PhD, start to measure which hand the baboon was using when he was communicating with his hand. And I remember having such an unexpected surprise that when the baboon were using their hand for communication, something happened in their hand preferences. You know, you can be right-handed or left-handed when you manipulate objects, but here, when they start using gesture for communication, the use of the right hand was exploding. It was very strange phenomena, and we were wondering like why. And even it was a case even in left-handed baboons. They were using more the right hand for communication.
And that is what Adrian and his student Yannick Becker have since gone on to study, confirming that they principally use their right hands when they make gestures and other signals, and that their brains too are asymmetrical towards the left, like ours is. So might this be a tantalising glimpse into the evolutionary origins of our own ability to communicate? Yannick takes up the story.
There's something peculiar about language in the human brain. It's processed more on the left side of the brain than the right. And when we want to study the evolution of language, we have to look at our primate relatives, our cousins, which in this case were the baboon, a particular species of old world monkeys. Adrian, my supervisor, has observed that these baboons use their right hand for communication more than the left hand. And the right side of the body is controlled by the left hemisphere. So because language in the humans is controlled by the left hemisphere, there was suddenly the hypothesis that maybe in these baboons it might be also the left hemisphere that is controlling communication, especially gestural communication.
That's intriguing, isn't it, to think that you've got that potential asymmetry going on in these close human relatives. Do they have right-hand dominance like we do, though? Because in humans, 90% of the population are right-handed, and the vast majority of those right-handers are controlling their right hand with the left side of their brain, which is also where language is, which is why we have this hypothesis about left-side dominance of the brain. Is that the same with handedness in these baboons?
It's very close. Actually, we divide handedness into two different ways of assessing handedness. So there's this, what naively we would call handedness maybe in the humans, that we use actions with our hands. For this, baboons are on a populational level a little bit right-handed. But then when we assess handedness for communication, special gestures that they make to communicate with each other, there are much more baboons right-handed for communicative gestures. And even those that have been left-handed in these manual actions will become right-handed in communicative gestures.
What sorts of gestures do they make then? How do they signal to each other with their right hands?
There's this hand slapping gesture. They slap one hand to the ground in order to intimidate. It's really a threatening gest, and if you see this, you immediately understand that this baboon tells you to go away. So there's a whole body posture that comes with it.
So is your sort of inference then that given that gestures are all about communication, and in humans a chief mode of communication is speech, and speech, we know, in the majority of us, is on the left side of the brain, that perhaps this is one step along the road to which our brains evolved the ability to speak because they had inherited this particular use of one side of the body for communication, and that we've taken it a step further.
That's exactly the point, yeah, thank you. We have inherited something on our evolutionary history from our ancestors, and this can be also found in these baboons. This asymmetry for this kind of communicative gesture might be a precursor of what we call language now.
So have you done the experiment you would do on a human? I mean, if we put a human in an MRI scanner and ask them to speak, we can see relative activation on the left side of the brain where they're creating that speech. Can you do a similar sort of thing in your baboons to see if they are similarly using that part of the brain asymmetrically when they're doing this communication?
Yeah, that's a bit the holy grail to get functional brain scannings. And it's very difficult to do actually in non-human primates. So what we've done is we're scanning these baboons, but they were asleep. So we have anatomical scans. And then our function would be the behavior that we have recorded.
And how does the anatomy compare left and right in the animals?
Actually, we can measure some regions of interest in both hemispheres, and then we can compare them and calculate what we call an asymmetric quotient. So we have done in this study this for a very special area that was shown to be an equivalent area to this famous speech production area in the human brain, which is called Broca's area, and compared in the left and the right hemisphere, and actually saw that this fold will be deeper in the left brain for those subjects that they were communicating with their right hand, and reversively, it was deeper in the right hemisphere for those that were communicating with the left hand.
So that sort of suggests that there is this specialisation that does seem to be associated with communication. But going back to the humans again, we know that about 10% of humans are left-handed, but they still do language a lot of the time on the left side of the brain. So did you find any baboons that bucked the trend like that and were lefties, but also appeared to have the specialisation for possible language still on the left?
Very, very interesting question. So We have variation in our data, but I think the most important point for us is that in humans we find this for this manual action handedness. And the hypothesis is that we would find this less if we assess handedness for communicative gesture also in the humans. But this has never been done yet. Would better predict in what hemisphere language is processed, or also in humans.
It's a fascinating story, isn't it? Yannick Becker there from CNRS. Sleep appears to be life critical. Deprive a human of it and they suffer metabolic and psychological consequences. Some animals even die if they're sleep deprived for long enough. And as most new parents know, given the chance, we catch up on long lost sleep by sleeping for longer later. But what about in the wild, where the pressure of daily life might not afford a sleep-robbed animal the opportunity to catch up like that? Do they go without? Well, that's what Carter Loftus has been trying to find out among Kenya's baboon population.
There's this homeostatic sleep drive. After animals experience particularly poor quality sleep, they respond to that by sleeping better the following nights, longer and more intensely. But in the wild, animals face a lot of competing priorities. Predation, they have to find resources, and we really don't know anything about how animals in the wild navigate between having to invest in sleep and social and ecological priorities.
So really the gold standard is then you're robbed of sleep, you make up for it in some way. But because we have very artificial circumstances when we're doing the sorts of studies that inform how animals and humans take sleep, we kind of don't capture that tension in the way we do those studies.
Exactly. The vast majority of studies of sleep have occurred in the laboratory or at the bedside, where these external factors that might shape our sleep are intentionally removed. But what they remove are the ecological and social pressures that may be really influential on the way that sleep has evolved in the environment in which it actually evolved.
Presumably part of the reason why those are not captured, though, is because it's actually quite hard to do this, I would think.
It is, and the gold standard for studying sleep research is polysomnography, which requires intracranially or subdermally implanted electrodes that can measure brain activity. And that's where we introduced a new method of using accelerometry to try to non-invasively answer some of these questions about how animals balance their need for sleep in the wild.
So accelerometry, that's basically capturing movement. So what are you using movement as a proxy for how awake an animal is? And therefore, when they're not moving, they must be asleep.
Exactly. We're essentially looking for periods of sustained inactivity. And then we're validating this measure of sleep against thermal video that we have of sleeping baboons in the wild.
Which baboons were you studying, and how do you capture that movement from them?
Yeah, we were studying a group of wild olive baboons in Laikipia, Kenya, and collared greater than 80% of the adults and subadults in the group with GPS collars that had an accelerometer unit implanted. And then these accelerometers collect essentially all the movements that a baboon does for the duration of the study, which was around 30 days.
That's powerful because that tells you what they're doing and where they are when they're doing it. So presumably you can also get the proximity. You can look at where they are in relation to each other as well.
Exactly. That's something that we looked at, as well as several other GPS-derived metrics that we were able to pull from the data, such as how far they traveled on a given day, where they slept in relation to their group mates, and how all of these variables actually had an influence on their sleep patterns.
The reason I brought that up is because obviously when we do sleep studies, often we divorce people from other stimuli, including each other. So being able to study how groups affect compensatory sleep or sleep behaviour is quite insightful. So were you able to capture that in this study?
Yeah, we were. And I think that was one of the most exciting findings of this study, is that the collective dynamics that animals experience when they live in groups really continue into their sleep period. And we found that the baboons that we studied actually synchronized their periods of nocturnal awakening, which is actually quite in contrast to a lot of predictions which suggest that animals should really stagger their periods of nocturnal awakening to maximize their collective vigilance and be able to hear and see predators when they're coming as a group.
So what did you actually see then? You track these baboons, you've got the whole lot over a really nice long period of time. So if there are periods when they're robbed of sleep, we should see compensation, etc. You've got the group dynamic. What trends and what patterns emerged which are not what we would predict based on previous studies of how individuals sleep?
Essentially, what we found is that animals in the wild can't afford to make up for lost sleep all of the time. The ecological and social pressures seem to be really the pressures that are driving their sleep patterns. So when they sleep in a new and unfamiliar environment, they sleep quite a bit less and they sleep more in a more fragmented way. And they also sleep quite a bit less when they are sleeping in proximity of more group members. But surprisingly, how much they've slept in the recent history and how much physical activity they had on the preceding day had relatively minor influence.
So presumably there's some kind of cost attached to this loss of sleep.
Presumably, yes, but we don't have a lot of data on how exactly those costs manifest. And the studies that have looked at this also found kind of difficulties in finding the exact cost of lost sleep, which kind of brings us back to one of the central questions of sleep research, which is, why do we sleep if we can give up sleep without major costs? And that's something that we still just don't really have a great answer to.
You don't think it's possible that in your animals where they have been robbed of sleep, that in fact they are catnapping here and there? I know you study baboons, not cats, but you know what I'm saying.
Yeah.
And that perhaps there is a bit of catch-up going on, but actually it's below the resolution of your ability to catch it with the accelerometer. So they're just dropping off periodically here and there throughout the day to compensate, and you're missing it.
That might be possible, but one of the beauties of accelerometry data is that we can collect continuous and really high resolution data for relatively long periods of time. So I would say that's rather unlikely. It could be that physiological need for sleep is playing out at timescales that we're just not looking at. Maybe they've given up sleep for 10 days or something, and then they're catching up on it in the next 10 days.
Carter Loftus, he's at UC Davis. To COVID now, and the COVID-19 pandemic is into its third year, as if I needed to tell anybody that. But the timeline means we're now in a position to look back longitudinally at the pandemic and ask how the virus is changing, as well as how our responses to it are also evolving. And when she was at the Fred Hutchinson in Seattle, Megan Garrett was interested in what sorts of antibody responses people were making, both following natural infection, but also in response to vaccination.
This was in the first days of people getting vaccinated against SARS-CoV-2. And we wanted to know, is the immune response that people make against the vaccine similar to the immune response that people make against the virus itself? And can the virus escape your vaccine-elicited antibodies in the same way that it can escape infection-elicited antibodies?
Did you suspect there might be a difference?
Yes, we suspected there could be a difference, mainly because people get infected and they don't get just exposed to the molecules on the surface of the virus. There's a whole host of things that come with getting an infection, whereas when you just get vaccinated, you're just given, in this case, mRNA to make the protein by itself alone.
So how did you pursue it then?
My original graduate project had been working with HIV, and in that context of HIV, we built a library of phage that display all sorts of little bits of the HIV proteins in this case. And then we can use that library and mix it together with antibodies, and you see where they bind. And right as I was publishing that paper with HIV, the pandemic hit. And so we thought, oh, let's build a library instead of against HIV, let's build it against SARS-CoV-2. And so this is a library that contains all of the little bits and pieces of the spike protein. But not only that, it also contains all the little bits and pieces plus one mutation. And so this is a library of all the possible single mutations for the spike protein on SARS-CoV-2.
You can therefore ask, right, when a person makes a response to the vaccine versus a person who's been exposed to the disease for real, what bits of the virus are their respective responses recognizing?
Exactly, yeah. And then our library also allows us to dive a little deeper and look at, okay, so this is where the antibodies bind, but what single mutations could cause a loss of binding? And that pinpoints what are the potential sites of escape.
And obviously that tells us something about where variants that can escape from the present generation of vaccines, the direction of travel in which they may head.
Exactly. It kind of shows the weak spots in our immune system and where the virus could potentially take advantage and mutate and escape.
Although, of course, there are going to be some changes which the virus would never be able to make, because were it to make them, it would cease to operate the way SARS-CoV-2 does.
Yes, exactly. So the one thing about our library is that it displays every single mutation possible, but it doesn't discriminate between mutations that are feasible, the mutations that could potentially exist on the virus, and mutations that could never exist because a mutation at that spot would just cause the protein to completely fall apart or not function or something. So it pinpoints potential escape mutations, but it's also important to do follow-up studies and see, okay, this mutation could cause escape, but can the virus ever actually make this mutation? That's the next step.
What actually emerges when you compare the response that people who have been vaccinated with the present generation of vaccines versus people who have been infected with the present generation of variants we've seen that you were testing here? What emerges?
We saw some really interesting differences and something unexpected. When we looked at vaccinated people, and we also happen to have a few samples from people that were severely infected. So these are people that were hospitalized. And we also had samples from people that had mild infection. What was interesting to us is that the binding patterns across spike between vaccinated people and severely infected people looked super similar, which was surprising to us. And if you look at people with mild infection, they have a completely different binding pattern. So for some reason, people that are vaccinated and people that have severe infection are making antibodies that bind to the same regions, which was really interesting to us.
And is that why people who are vaccinated, we are seeing as a general trend, might still get infected, but they 90% of the time don't get severe disease?
I don't know if that's directly why. Our hypothesis is that when you get severely infected and when you get vaccinated, you get exposed to just a ton of antigen. You're just flooded with antigen. And so your body is, when your body makes antibodies, it's given a lot more antigen. And so it'll make antibodies against like different regions than it would if it was just given a little bit of antigen. And that's just our hypothesis, because that's kind of the similarity between people that had severe infection and that were vaccinated. They're just given a ton of antigen.
One of the things that researchers are beginning to talk about more is COVID vaccine 2.0, as they're dubbing it. The idea that we could come up with some kind of pan-coronavirus vaccine by finding the parts of the outer coat which do appear to be invariant across the strains and variants that emerge. What do you think the prospects of that are? And did you find any areas that would be perhaps good contenders to try and reinforce an immune response against those particular areas?
Eventually, if we're not doing it now, we're probably going to be moving towards creating a vaccine against more conserved regions. This is kind of what's happened with flu, where people are trying to target the stalk region, which is a lot more conserved. And it's very similar to the S2 region on spike, which is also very conserved. It has a lot of the same functions. And so it's taken years for the flu field to kind of move towards, oh, we should be vaccinating against these conserved regions. And I think we've realized that kind of earlier with SARS-CoV-2. So we kind of needed these vaccines quickly and they worked great, but we're now thinking more about, okay, let's do this in a more considered way. And I think that'll be the next generation of vaccines.
Megan Garrett. As a child, I can remember standing transfixed at the London Natural History Museum in front of the remains of a massive armoured dinosaur. This was one of the group of animals that were covered in thick defensive plates that basically turn them into living tanks. Well, now a discovery in China brings us a bit closer to the root of how those animals originated and then disseminated worldwide. Shandong Bi set out originally to look for ancient mammal remains, but a chance meeting in a village sent him off in a very different direction and led to the recovery of the new species we now call Yuxisaurus kopchiki.
You can imagine this is kind of like a armadillo, right? But it's much, much bigger in size, about like 3 meters long, and it's covered with a thermal plate over the body.
Basically, it's a small car. Is that a reasonable comparison? That sounds large.
It's like, I say, like a truck.
And when would this have been around?
This group include Stegosaurus and Ankylosaurus. So it's around from like early Jurassic to the late Cretaceous. It's roughly 190 million years to 70 million years ago.
Would it have weighed as much as a truck? If you'd put one of these things on the scales, what would its weight have been?
Probably like 1 ton.
So it is sort of car mass as well, isn't it? What did they eat? Do we know much about their diet?
Based on the teeth, they probably is a herbivore. Leaves of plant and some like a juicy fern.
So it's basically elephant in terms of size, for comparison. It's going to eat like an elephant as well and weighs as much as a car. I mean, it's a pretty big thing. How did you find this?
Actually, I'm a mammal specialist. I was looking for the first mammal because we believe the first mammal was found in southwestern China, like Yunnan province. So I led the team to a site 100 kilometers west of Kunming, the capital city of Yunnan province. I went to the site called Jiaojiadia village, and the local people showed me osteoderm, the derm plate. I was impressed. I said, oh man. So we went out to the field, and from the same site, then we dug out more than 100 bones, including skull, lower jaw, and the vertebrae.
So are you saying that the locals had already found this, or at least they'd found some specimens that were one of these, and so that gave you the clue that there were these specimens in the area?
Yes. The area also very rich in fossils. They already, before, they also found a lot of species like a sauropod, the long-necked dinosaurs, and also some theropods.
And the dating for this, did you get the dating from the stratigraphy, in other words, the rocks in which it's found, or did you date it by finding out what you got and then comparing it to other things to work out where it must sit in the timeline?
For the dating, it's mainly based on the rock sequence. We make the lateral comparison with the nearby locality called Lufeng locality. At that locality, they already did dating, use geochemical, and also based on stratigraphy. So we kind of confidently say this early Jurassic.
Just so we can appreciate, where does that sit then in the timeline of these sorts of dinosaurs? Is that really early then in their evolution?
I would say it's really early, like the beginning of the Jurassic, really early.
And so this is the sort of ancestor that would have become the big beasts like stegosaurs and so on, that we're very familiar with playing with toys of them when we're little. Is that what you're saying, that this is probably the ancestor of some of those very big herbivorous dinosaurs that were plated and well defended?
Yes, the armed dinosaur include the most famous group of Stegosaurus, Ankylosaurus. As you familiar, Stegosaurus is a plate along the back. Ankylosaurus is a derm plate all over the body. For this new species, it's a primitive members before the Stegosaurus and the Ankylosaurus split. So it's the ancestor of Stegosaurus and Ankylosaurus.
Obviously, if they've got that level of defence, they must have it for a reason. So there must have been around at the same time things capable of penetrating that sort of armour to want to attack them and eat them. Have we got some insights into what was eating these animals?
we are uncertain, what species ate this group because you look at these creatures, they are fully covered by the dermal plate. I don't think it's, many creatures was larger than this.
And has anyone found anything like this anywhere else? You've got this from this particular part of Asia, but have you got anything like this anywhere else?
A similar species has found in Britain and in Germany. So we are unsure, that where, which continent this group originated. But what we can tell based on the new discovery, we can tell, in early Jurassic, this group already spread very rapidly around the world.
Shandong Bi, he's at Indiana University of Pennsylvania. And that is where we must leave it for this month. You have been listening to the eLife podcast, which is produced by The Naked Scientist. There are previous editions, references, and also the full text transcripts for these programmes and details on how you can subscribe to this podcast at nakedscientist.com/eLife. And just to let you know, The Naked Scientists also produce a weekly science programme which covers the latest leading science stories. You can find that at nakedscientist.com/podcasts. I'm going to be back at the same time next month with another look inside eLife. Until then, from me, Chris Smith, thank you very much for listening and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.